Okay, and I'll go. All right. Good morning, everyone. How is everyone today? It is an honor to present Xin Lin today, who is this year's recipient of the Grace Marie Kaler Endowed Award in Cardiology. We're joined by Ms. Kaler's son, John. Thank you, John, for being here today. The Division of Cardiology awards this award each year to an outstanding presenter of our monthly research conference, and this year, Xin got the award. As you all know, Xin is an assistant professor in our Hartford section and a true colleague. His training and education reads like a geographic map of the finest institutions in the United States. After attending college at Harvard, he completed an MD PhD at Johns Hopkins with a thesis on methods of gene mapping and simultaneously obtaining a master's in biostatistics. He trained at Mayo Clinic at University of Pennsylvania before doing his cardiology fellowship at Stanford and a postdoctoral fellowship in epigenomics. He then completed a Hartford fellowship at UCLA. With his experience on the West Coast, we're glad he decided that the West Coast is the best coast, and I decided to stay here and join us at the University of Washington. He has received numerous awards and is prolific with 38 publications focusing on genomics. In his early career, he has already received an R00 to study the epigenomics of dilated cardiomyopathy. Today, he is going to talk about slightly different research interests on the genetic studies of electrophysiologic traits. Please join me in welcoming Xin Lin.
Okay, thank you for that warm introduction. Because of the time constraints, I might have to talk a little faster today. Okay, so I wanted to motivate the subject of today's talk with a case that I recently encountered. It's of a 71-year-old male with stage D class 4 heart failure on palliative milrinone. He got some VT as an outpatient and was ATP'd out of it. And he was slated to come in for amnio loading. And he said to me, "Doc, I don't really want to come in the hospital. This medication may help me live longer, but you have to understand in 2 or 3 months I'm going to enroll in hospice anyway." And what I replied back to him was that amnio is not going to extend your life, but it will decrease the chances that you're going to get shocked. And that's a worthy pursuit no matter what goals of care you have. And so I just want to emphasize the point that although ICDs save lives, anti-arrhythmic drugs still have a place in how we manage arrhythmias. And frankly, if we had more of them to use, that would improve patient care. It turns out that a month later he had VT arrest at home, and he had ACLS, and ROS was achieved. He had a lot of pain from the chest compression, so he was made DNR DNI, and he was recovering well, but he had more VT in the hospital, had multiple ICD discharges, but without ACLS he expired. So this all just shows that ICDs are great, but it's not perfect, and there's more work to do in the field. Today I wanted to highlight on a high level the history of antiarrhythmic drug development. I want to talk about the basic science that was the impetus for antiarrhythmic drug development. I want to talk about the current atmosphere in which interest in the area has sort of petered out, and suggest that genome-wide association studies may be a way forward in the field. And last, I want to talk about my own work in the field of the genetics of electrophysiologic traits. Okay, so when I first looked into this topic, I just assumed that most of the electrophysiology drug armamentarium was simply grandfathered in from anecdotal usage, anecdotal efficacy. But it turns out that most drugs were actually formulated from a rational drug design concept. And there's only 2 medications that got grandfathered in, and that's quinine, which became quinidine, and digoxin. Everything else came out of rational drug design. And it's not the sort that we're used to today with 3D structures and different moieties and things like that. But it was rational for the technology that they had at the time. And this is the technology. It was The whole theory behind it came out of work by Hodgkin and Huxley. Here is a experimental setup in which they've put an electrode in the axon of a squid nerve, and they've measured the action potential here. And for their work, they were awarded the Nobel Prize in 1963, and this is the Nobel Prize program with them there. And apparently they also experimented on lobsters, poor guy. But this is the equation that they put forth to model what they observed, and it involved a term for sodium channels and potassium channels, and also a leak channel. And the action potential would go off at the application of an external current. So then let's move from the science of a nerve axon to cardiomyocytes. And initially, scientists thought that they could essentially take the Hodgkin-Huxley model wholesale and just tweak it a little bit for cardiomyocytes. And so in 1960, that's exactly what they did. They added a sodium rectifier channel. But in 1964, as the technique was being refined, they performed careful patch clamping on cardiomyocytes, and they found that, in fact, there were other entities involved in action potential production in cardiomyocytes, such that in subsequent years, researchers devoted themselves to trying to find what those other components were, and calcium channel and other exchangers were found subsequently. Now, it is upon this scaffold of different channels and exchangers giving rise to the action potential that anti-arrhythmic drug development occurred. In 1963, Vaughan Williams noticed that the first anti-arrhythmic drug decreased the rate of the action potential upstroke by affecting sodium channels. And that led the field to look for other compounds that would have an antiarrhythmic effect through that mechanism. And then they noticed that among the drugs, another group, a portion of them had a beta blockade effect, and so that became class 2. One beta blocker in particular also had antiarrhythmic effects by increasing the action potential width through its effect on potassium channels, and that's sotalol, and that became class 3, the beginning of class 3, so on and so forth. And so there's not a one-to-one relationship between the scientific discoveries and then the classes of various antiarrhythmics, but I think it's fair to say that had these discoveries not been made, then certainly antiarrhythmic drugs would not, the development would not have followed this course if it would have occurred at all. With this scientific framework now developed, it was really off to the races in the field, so much so that in 1983, Brahma Singh, who is a giant in the field, wrote at the beginning of one of his papers, although a bewildering plethora of new antiarrhythmic compounds has recently been developed and characterized, etc. So this is really a blossoming of antiarrhythmic work, discovery, and therapy. So anybody with any situational awareness knows that the atmosphere now is completely different. The last antiarrhythmic drug to be approved was in 2009, and that was dronedarone. And really interest in the field has waned. And the reason is that there were pivotal studies that came out, the CAST and SWORD trials, that showed that antiarrhythmics not really decreased lethal arrhythmias. They actually contributed to mortality, especially in the context of MI. Now, to all the meta-analyses that have been done to date, no study's ever shown that an antiarrhythmic has extended life, except beta blockers. But I think most cardiologists would consider beta blockers not true antiarrhythmics in the sense that you would use them for chemical cardioversion. And in the last 10 to 20 years, most of the innovation in the field has been on device and ablation therapy. And in reviewing this history, I got the sense that this scientific framework of channels and exchangers had taken us as far as we could go in terms of antiarrhythmic drug development. I'm not the only one who felt this way. The Sicilian Gambit is a collection of experts in the field. They met 4 times. They're best known for revamping the Vaughan Williams Singh classification. And they did so wanting to, quote, inspire and direct research, development, and therapy. Now, we all know that their construct largely has been abandoned, but they did make a number of noteworthy observations. And in their last meeting, they wrote that not only to promote antiarrhythmic drug development should we look at the ion channels, but we need to look at modulators of those ion channels and effects on electrophysiology that have secondary effect like remodeling. And so to really propel the field forward to increase interest in antiarrhythmic drug development, there's this notion that we need to find new components that affect electrophysiology of cardiomyocytes. Okay, so I'd like to next talk about genome-wide association studies as possibly a way forward to introduce new components in our understanding of electrophysiology. Let me explain what this is. Genome-wide association studies, they're quite versatile. One can study almost anything with very little prior knowledge, so long as one can measure the phenotype of interest in a group of people and then get their DNA. And then so you go type them, the cases and the controls. In this cartoon example, then you literally count, how many variants are in the cases versus the controls, so that in this example, 7 of 12 have the red variant among the cases, and 2 out of 12 have the red variant in the controls. And then you can then posit, because there's this difference, that this locus is associated to your phenotype of study. Now, before I get heckled out of here, clearly this is a gross oversimplification. In real life, each person is diploid. We don't have just one chromosome. We have 23 pairs. And to correctly power these types of studies, we need not dozens of individuals, but thousands or tens of thousands of them. And we're not looking at just one site of blue and red. We're looking at millions of sites. So this is a more realistic depiction of how these studies go. And you don't just need to look at something binary like AFib and no AFib. You can also measure a continuous variable like height or weight or blood pressure. You can even look at something abstract like the expression of your favorite gene. And what you do is you have your phenotype measured in a lot of people, and then you look at many variants, and at any single variant, there's usually 2 alleles. And that's just a fact that the vast majority of variants in the genome have 2 alleles. Theoretically, they could have 4, but there's some population genetics underpinnings that can explain why the vast majority of them have only 2 alleles. And then so for any variant, then you can make 3 groups, the 2 homozygotes and the heterozygote. And you can see if there are any differences among the different groups for your phenotype of study. And in most places, there won't be. But in some there will be. And so when geneticists talk about an association between a variant and a phenotype, they're talking conceptually about making plots like these and seeing if there's differences between the groups. And if there are, then you can assign a p-value to this plot. And if you take the negative log of the p-value, then the higher the value, the more significant it is. And then you can go along the genome here and plot these values. And in most places, it'll look like this, right? There's just going to be no relationship between your genotypes and the phenotype of study. But in several places in the genome, there will be an association between the SNP or the loci and your phenotype of study. It'll look like this. Okay, so one area of active work has been GWAS of EKG parameters. You can use PR interval, QRS duration, QT interval as your phenotype. This is relatively easy to do because EKGs are relatively easy to get. They're not that expensive and they're non-invasive. And so there have been a lot of studies conducted for those type of phenotypes. Of 2,313 entries in a public compendium of GWAS studies, 23 studies have been performed on EKG parameters with 236 loci implicated. And so at this point, you might be saying to yourself, well, with all these loci associated to electrophysiologic traits, where's the drug development? Well, the problem is that there's an obstacle hindering the field, and that is what I'm going to try to illustrate now. So you have the most significant GWAS hit, say it's right here, somewhere in the genome, and then you got genes all around. It's not clear what this SNP is a surrogate for, which gene it is, okay? And without a gene, it's difficult to do downstream mechanistic studies and find druggable targets, etc. So this was mentioned in an expert in the field 2 summers ago, Terry Manolio, who wrote, associations identified by GWAS still explain only a small fraction of the heritability of complex diseases, and the overwhelming majority fall in regions of the genome that have no known function. So again, if the SNP is in no man's land, no genes on top of it, then it's hard to move on. So let me talk about my notion of how to solve this problem. And I have to give credit to Nana, who's at the CHRU in our division, who helped me formulate the concepts of this research. My notion is to use GTEX data to help pick the trait gene. And so what is GTEX? It's one of the offspring of the Human Genome Project. They took 1,000 individuals, did whole genome sequencing on them, and then took different tissues and did RNA-seq on them. Now these are considered normal, so clearly these samples came from deceased individuals. As of their last data freeze, they have 433 left ventricle samples and 429 right atrial appendage samples. Okay, so how does GTEX data help us in choosing the trait gene? So this is the problem again. We don't know which trait gene to choose. So now this most significant GWAS hit, by definition, is associated to your EKG phenotype, right? That's what all those past GWASs have done for us, is given us this information. Now, there's some gene in the locus that's associated to the EKG phenotype as well. Okay, we just don't know which gene it is. But since there's an association between the SNP and the phenotype and the expression of a gene in the phenotype, then by transitivity, what we can look for is the association between the SNP and the expression of the genes around the locus. Okay, so you conceptually, what you do is you make these plots for all the genes, okay, in a given locus. And what I do is I have a 1.5 megabase window, and I make these plots for all the genes. And then if there's a difference, then we can say, this is likely the gene that is giving rise to the phenotype of study. Okay, so let's make this concrete. This is very difficult to see, but I'll walk you through it. This is one locus that has been associated to an EKG parameter, to QT. So this is the SNP, and this is the p-value of the association. And you might be like, well, that's not genome-wide significant, and you're right. But investigators looked at a SNP close by that was correlated to that gene and replicated it with this p-value. Now you also might say, well, this p-value is not as significant as this one, but the issue is that for this was a genome-wide association, so you have to correct for genome-wide numbers of other alleles. But here, the replicate cohort, they were only looking at a handful. So this is actually a bona fide association. Now, these two SNPs are sitting right in this gene called NUX1. And so, absent of any other information, if you had to choose a trait gene from that finding, I think choosing NUX1 makes sense. But when you look at the expression of the gene with respect to the different genotypes, you'll see that there's no relationship. Okay, so then we can try looking at the expression of other genes around there, and lo and behold, there's a gene here, PM20D1, which with respect to this SNP has this relationship in terms of its expression. So, if you're a betting person, you probably want to bet on this guy giving rise to your phenotype and not this one. So that's one locus. I've done the same thing to 171 other loci associated to atrial-related EKG parameters and 278 loci associated to ventricular-related EKG parameters. I've used the GTEX data on them, and GTEX is informative only a fraction of the time, but it's still a good number. And surprisingly, the candidate trait gene I pick by using the GTEX data is the vast majority of the time discordant from the one that authors reported in the original studies. This is due to some extent because authors sometimes didn't report anything because they couldn't figure it out. The GTEX candidate trait gene is oftentimes not the gene closest to the SNP that was most significant in the GWAS. And the final thing to say is that oftentimes authors also did not choose as their candidate trait gene the one that was closest to the most significant hit. And so what we're left with is three overlapping but somewhat disparate groups of candidate trait genes. One that you can pick from looking at the proximity to the closest that's closest to the most significant hit. Another by what authors chose, presumably by looking at the genes around there and reading about the literature and picking one that fit. And finally, one that is more data-driven using GTEX data. And so then the question is, can we assume that this group is the best one because it's more data-driven? We actually need to do validation, and probably conceptually the best way to do it would be to knock out all these candidate trait genes in mice and then do EKGs on these mice. And actually, Nana did that for one candidate in one of her papers. But we have hundreds of candidates, and so this is just not going to be practical. We would have to have like 300 different colonies of lines to do that, and I just don't think there's that much space at the bottom of South Lake Union to handle that. So we need some higher throughput way to validate these candidates. And so I've teamed up with Ken Zhang and Deok Kim to put together this high throughput validation system. We're going to take iPSC cardiomyocytes, so these are induced pluripotent stem cells, and we've differentiated them into cardiomyocytes. And then we're going to take siRNAs and knock down the candidate genes. So this is a technology where you add these agents and then the expression of your gene of interest is knocked down. And then we're going to look for expected perturbations on MEA, multi-electrode array, so that, like, you know, if we go back to that example with NUX1 and PM20D1 that was associated QT, we're going to knock down those specific genes on these cells and look for changes in the QT on a multi-electrode array. So let's talk about multi-electrode array. It's really just an EKG system for cells in culture, and this is one MEA. And you can see that the readout is kind of like telemetry we all recognize. They have their own lingo for certain things. Instead of talking about QRS interval, they talk about spike slope. And for them, QT is field potential duration. And then you can measure action potential across the lawn of cells, and that'll give you the conduction velocity. The nice thing about these MEAs is nowadays you can buy off-the-shelf systems that are in 48, 96 well formats, and so you can do a lot of testing easily. Now, I've so far sort of glided over this issue, but a known problem in the field is these iPSC cardiomyocytes. The problem is that people know these cells are very immature, maybe even less mature than fetal cardiomyocytes. And so how do we get these cells to be more like adult cardiomyocytes, like the real thing? So Deok Kim's notion is to have them sit on top of a nanopattern surface. And a nanopattern surface is really nothing more than a collection of parallel grooves. Now, why would this mature a cell? It's, in theory, supposed to mimic the basement membrane during development. And so he has these cells sit on top of this construct for a while, and then he puts the whole thing on the MEA. Now, I just wanted to show you some evidence of maturation of these iPSC cardiomyocytes. So on a default flat surface, this is what cells look like. They're sort of disorganized, they're all over the place. But on a nanopattern surface, they acquire this more linear organization. And I actually showed this to Dr. Shu in pathology, and he thought this came straight out of a person's heart. And I said, no, these are actually iPSC cardiomyocytes atop a nanopattern surface. And so morphologically, cells treated in this fashion look like the real thing. Now, there's other molecular evidence that the cells are more mature, but, you know, in the sake of time, I'll just sort of bottom line it for you and just get to the end. The question is, do we really need these more mature cells to manifest genetic perturbations, right? So in this plot, combo mat refers to various maturation cues, including the nanopattern surface. So for the purposes of this plot, just think nanopattern surface when you see combo mat. Now, we're working with two lines, one wild type and one in which we've knocked out Duchenne muscular dystrophy gene. Actually, we've knocked in a known mutation. Now, people should know that that disease manifests VT. Now, on an MEA, we actually don't see VT. Instead, we measure a surrogate change of beat interval. And so when you look at the wild type, both control and on combo mat, you'll see that the beat variability is not really remarkable, as we expect. And then with the mutation, DMD mutation, on default flat surfaces, you also don't see anything. But with the combo mat on nanopattern surfaces, then you see that the expected phenotype is manifest. So it really is important that we use the most advanced iPSC cardiomyocytes as possible. So let's go back to that chromosome 1 locus between NUX1 and PM20D1. We looked at the QT after knocking down NUX1 in one set of cells and then PM20D1 in another set. And you can see that the NUX1 doesn't seem to have an effect on QT, well, at least not as profound as PM20D1. So at least for this example, it seems that the GTEX data really did help us choose the right trait gene. For sticklers out there, there's no p-value around here because the data were unbalanced and I wasn't able to analyze it in the correct way, but stay tuned. The last thing I wanted to say was that, I've set this talk up sort of as a competition between different groups of candidate trait genes. But in actuality, if you look at the actual genes in each group, you will see that the vast majority of them are not recognized channels, and they are not in any way known to be connected to cardiomyocyte electrophysiology. And so whatever genes come out on top, whichever group, we're going to be validating and bringing in many more genes to implicate them in cardiomyocyte electrophysiology than we would have before the study. And hopefully that'll lead to more mechanistic elucidation, greater understanding, and more drug targets in the future to spur a second wave of anti-arrhythmic drug development. Okay, so in conclusion, hopefully I've conveyed to you what's going on in anti-arrhythmic drug development field, how GWAS can help perhaps propel the field forward by finding new components involved in electrophysiology, that GTEX data may help us bridge GWAS loci associations and actually identifying trait genes, and these advancements in bioengineering using iPSC cardiomyocytes matured on NP surfaces atop MEAs is a high throughput means to validate candidate trait genes. I'd like to thank Arun, a postdoc in my lab, for all his hard work, and other individuals here, and open it up for questions.
Thanks, Jim. Great talk. I have a question for you, and it stems from a lot of these sort of in vitro or attempts to try and identify. You started the talk with a case with arrhythmia, went to in vitro. How are you going to validate that arrhythmia part? Is the mouse an adequate model? Is there some other mechanism to sort of take what you're doing in culture and get some idea of whether it is valid without using humans as your...
Right, yeah. No, this is a great question. So far I've really just thought about this topic in trying to generate new mechanisms in electrophysiology, just as a basic science question. In terms of its application to actual disease, I think that's going to be a much more downstream. Hopefully some of these will sort of call out for certain or be obvious for certain arrhythmias, and then we could use them as targets for drugs and things like that. But right now, absent of any further downstream work, I think it's hard for me to conceive of something like that.
And you also suggested a lot of what you found from the screen wasn't classic channels.
Yeah, that's right.
So how are you envisioning all of these, like what were those other classes of genes, and how are you, like, you've got the data, but it's like, what's the model of how they are actually leading to the changes?
Yeah, so a lot of them, like, you just look them up and there's like blank pages. So like, they're just completely not described or not known genes. I think that's the excitement of really coming up with new concepts. Some of them are more like, it's more obvious, like there's like a phosphokinase or something. You can imagine that phosphorylating a channel. But there are a lot of genes that nobody knows anything about. Nana?
Fabulous talk, very highlighting some of our important limitations in GWAS. I really liked it. To answer a quick question for Rob, one of the big groups of genes that are coming out of transcription factors, particularly for the conduction phenotype. So things that influence, particularly cardiac transcription factors, things that influence cardiac development. Question for you, and I know you know this well, that tissue is really important. For example, whether it's a atrial myocyte versus a ventricular myocyte versus whether even it's a Purkinje fiber. So going to your the cells that you are developing, what are they? Are they mostly ventricular myocyte? Are they what are they, and how can you influence them to become even more subspecified?
Yeah. So there's no, when people talk about iPSC cardiomyocytes, it's not a monolith, right? Like the ones that you buy from CDI, which are high quality, they're actually a mixture of ventricle, atrial, and nodal. So that's not the best thing. So we're actually looking at ways to try to further differentiate these cells into ventricular-like, atrial-like, nodal-like, and that's by application of retinoids. So we're looking into that, but and that's a direction we definitely want to head into, and it's important because the loci that have been implicated for atrial EKG parameters are disparate from the ones for loci associated to ventricular EKG phenotypes. Go ahead.
I'm interested whether the produced proteins from these genes affect structure or enzymatic proteins. And I think both types of proteins could be relevant here, but where do you think the emphasis is going to be, or on both?
Yeah, so the exciting thing is that a lot of these guys are going to potentially not be channels at all, and so how they're going to affect, come down to affect at the very end these channels, we're just going to have to see. This is a sort of omic project, looking at the forest, not any particular tree, but it will be exciting to look at what genes get validated and then see if there are any trends, but we're not quite there yet. Alina? Sorry.
We're going to make that the last question.
Yeah. No, this is a known thing that your iPSC cardiomyocytes, what line they're derived from matters. Like some are more amenable. We're just using default WTC11, which sort of behaves well for this application. But you're right. It's that the line which it came from does affect the results.
So I think all of you can see why Dr. Shinlin is this year's recipient of the Grace Marie Kaler Endowed Award in Cardiology. Congratulations.
Thanks.
So the division awards the recipient each year for an outstanding presentation at our monthly research conference. And John Kaler is here today, and I'd like to ask you to step up and join us. So I'm going to read what this plaque that you're about to get says, and then I'll ask John to just say a couple words. So it states that throughout her career, Grace Marie Kaler worked in research laboratories across the University of Washington. She was known for her expertise and for the way she shared her skills and talents with the trainees, helping them improve their research techniques. This award honors the impact Mrs. Kaler had on many lives, and it preserves her legacy at the institution she served with such distinction. So, John, if you have a few words.
Just I thank you all for helping to keep my mother's memory alive. She came back to work here when I was in about third grade, and she worked up until the 90s, and it was fascinating. Every day she'd come home with a story about things, and we'd sit there and watch Nova and all. Anyway, so this is like watching Nova again. I really appreciate it. But I was thinking about it, what you were saying about my mom and how she would pass on techniques and things. That was one of her big things. Is after she retired, she went and started volunteering at a local school near her house, and the kids all knew her as Mom K, and she's always loved to teach people. And probably none of you knew my mom, but maybe some of you learned a technique that was taught by someone who learned it from my mom some years down the road. So anyway, but thank you very much, and that was a very outstanding talk. Thank you.